All right, welcome back to The Explainer. Go ahead and grab your lab coats because today we are stepping into the role of exam detectives. We've got a seriously fascinating challenge straight out of the WAEC 2026 chemistry archives to crack. And hey, a quick shout out to QandAHelp for powering today's investigation with their reviewer-checked past questions. Let's dive right in and see exactly what kind of chemical mystery we're dealing with today.
So, here is the scene of our chemical crime. WAEC 2026 Chemistry. Question 4. The question asks, carbon 4 oxide could be produced from glucose by, and then it gives us 4 totally distinct processes to choose from. A. Decarbonization. B. Oxidation. C. Fermentation. Or D. Hydrolysis. Now, what's really interesting about this setup is how it immediately presents a puzzle for us. We have a starting material, which is glucose, and a final product, carbon 4 oxide. Our job as exam detectives is to figure out exactly which of these 4 routes represents the targeted process that actually bridges the gap between the two.
To make this perfectly clear, let's just strip away all the extra noise and look at the core chemical transformation. We're starting with C₆H₁₂O₆. That's our glucose, a relatively large, energy-packed molecule. And we need to get to CO₂, carbon 4 oxide. There is a missing link right in the middle of this equation. A very specific chemical process that takes that complex sugar and breaks it down to release those CO₂ bubbles. Honestly, visually isolating the formula exactly like this is a fantastic way to train your brain during an exam. You aren't just reading words, you are looking at a biochemical journey. What is the vehicle that takes us from the left side of this arrow all the way to the right?
Inside the yeast factory, breaking down glucose.
Let's explore the underlying biochemical concept here by looking at a pretty amazing natural process. I want you to think of yeast as a microscopic, incredibly efficient chemical factory. When yeast enzymes get to work on a glucose solution in an environment with little or no oxygen, they perform a process called fermentation. The yeast essentially eats the glucose, processes it, and then literally breathes out 2 very specific things. First, ethanol, and second, our target molecule, bubbles of carbon 4 oxide. And this just brilliantly illustrates the exact transformation our WAEC question is asking about.
When we actually map this out as a balanced chemical equation, we can see exactly what happens. Over on the left column, we have our reactants, right? C₆H₁₂O₆, which is glucose. On the right column, we have our products, which gives us 2 C₂H₅OH, that's ethanol, plus 2 CO₂, our carbon dioxide. So the absolutely crucial point is right there on the right side of the equation. Carbon dioxide is a direct, explicitly stated product of this reaction. When you see glucose turning into ethanol and CO₂, your exam reflexes should immediately scream fermentation.
Next up, interrogating the suspects, ruling out the wrong answers. Now, let's put our detective hats back on because we definitely have a very suspicious character in our lineup, and that's option B, oxidation. For the highly observant chemistry students out there, you might be thinking right now, wait a minute, complete oxidation of glucose also produces CO₂ and water. And scientifically, you are absolutely correct. Complete oxidation of glucose, so that's C₆H₁₂O₆ plus 6O2, does indeed yield 6CO₂ and 6H2O. But here is the critical exam nuance you need to catch. In the context of this specific WAEC-style question, they are heavily targeting the classic yeast-driven conversion of glucose into ethanol and CO₂. While oxidation is a scientific possibility, fermentation is the definitive expected answer they are looking for to test your knowledge of this specific organic breakdown. So what about the other suspects? Let's quickly eliminate them using some basic chemistry logic. Option A is decarbonization. Now, this simply refers to removing carbon-containing components from a substance. It's completely unrelated to the specific biological yeast conversion of glucose that we are looking for here. Then we have option D, hydrolysis. Hydrolysis involves using water to basically split large, complex compounds into smaller ones. But glucose is already a simple sugar, it's a monosaccharide. It just doesn't fit the profile for the CO₂ producing process we need. So both A and D are cleanly ruled out. And that brings us to our grand reveal. By process of elimination and by really understanding the chemical equation, we can confidently declare option C, fermentation, as the correct and expected examination answer. The mystery is solved, guys. The breakdown of glucose by yeast enzymes perfectly bridges the gap between C₆H₁₂O₆ and the production of CO₂ gas alongside ethanol. Before we test your reflexes on this, let's lock in a golden rule, a really good shortcut for our short-term memory. Just remember this catchy little phrase: yeast eats glucose and releases ethanol plus CO₂. Honestly, whenever you see these keywords in a WAEC, JAMB, or NECO exam, glucose, yeast, ethanol, and carbon-4 oxide, you can almost guarantee the concept they are testing is fermentation. Make sure you lock that quote in.
Moving on to flashcard reflexes, chemistry exam quick quizzes.
Okay, it is active recall time. Let's see just how fast your flashcard reflexes are. Here is quick quiz number 1. During fermentation, glucose produces ethanol and which gas? Is it A, oxygen, B, hydrogen, C, carbon-4 oxide, or D, nitrogen? Take a second and lock in your answer right now. And the correct answer is C, carbonate oxide. Great job if you got that right. This basically reinforces that critical, unbreakable link in your mind between the process of fermentation, the byproduct of ethanol, and the release of carbonate oxide gas. They always travel together in this context. All right, let's step it up a notch for quick quiz 2. This time, we are looking at the actual balanced equation. Complete the equation. C₆H₁₂O₆ yields 2 C₂H₁₆OH plus what? Is it A, 2 CO₂, B, O2, C, H2, or D, NH3? Think back to the exact balanced components we discussed earlier. Have it? Lock it in.
And the answer is A, 2 CO₂. Now, what's really important to remember here isn't just the CO₂ part, but the coefficient 2 sitting right in front of it. That 2 signifies that 2 molecules or 2 moles of CO₂ are generated for every single mole of glucose that gets broken down. That is the absolute hallmark of a perfectly balanced fermentation equation.
So, my fellow exam detectives, you have officially mastered the chemistry of glucose fermentation. You know the reactants, the products, the balanced equation, and exactly how to navigate tricky distractors like oxidation. But I want to leave you with a final thought to chew on today. If yeast can do this naturally without oxygen, what other invisible chemical factories are operating all around us right now? Mastering one question is great, but understanding the chemical world is the real goal here. To continue conquering your WAEC, JAMB, or NECO syllabus, head over to QandAHelp.com right now to tap into a massive library of organized, reviewer-checked past questions and in-depth explanations for every subject. Continue your study streak, stay curious, and I will see you in the next explainer.